Welcome to Human Physiology. So we're going to start with Chapter 1, the Introduction to Physiology. The way your textbook is organized is it will give you an overview over what this chapter is about. And so today's chapter will be just talking about what physiology is and how we're going to study the function of the human body. The themes in physiology, the most important one that you will find sort of like a red thread going through the entire course is homeostasis. So that's definitely something that you need to understand what that is. We're going to talk about it in this chapter. We're going to talk about the control systems and how they achieve homeostasis. And then, of course, physiology is a science. So ology is the study of, and physio would be the body. In our case, would be studying the human body. And I have some pictures here for you. You can take a look at some amazing things that some athletes can do with the human body. So it's always some pretty pictures to look at. And let's get started here with physiology is an integrative science. That means that it's not enough to just understand the parts individually, but you need to understand how they work together. In anatomy, you were studying a lot of all the different parts and you got to name them and everything. But in physiology, knowing the parts is not enough. You need to be able to understand how one system affects another and how we have all these different levels of organization and how really they all work together to achieve homeostasis. We won't be talking too much about the term genomics or the Physiome project. So genomics, a lot of people are into the sequencing the whole genomes of organisms and then compare them, but it's not going to be much part of this course. The levels of organization. So physiology defined. Again, ology means study of, and physio, the body, in our case, the human body. And this would be including all of its chemical and physical processes. So let's take a look first at sort of the organization of life from the very smallest units to the very larger context. And there, well, you can start with chemistry, and there you have atoms, the smallest unit of matter, and then molecules, cells, tissues, organs, organ systems, and organisms. Let's take a look at where physiology comes into play. That'll be this slide right here. You can see that physiology spans quite a range. So here, this is sort of the biggest circle that you have here, a half circle. We're starting with molecular physiology, and you will take a look at some of the molecules that are inside of cells. And that's important. That's sort of my expertise. I'm a molecular physiologist, so I was always looking at sort of the inside of cells and how they work. But then in physiology, you just want to move on also to tissues, organs, organ systems, the whole organism. And you may even include how organisms work together in populations. Anyways, so here, let's take a look at the organ system review. We have 10 systems that we'll be going over in this course, and they're listed all here. We're going to have an introductory unit that talks first about things like diffusion and a little bit on genetics, protein synthesis, and some other general biology topics, I would say. But then we'll be going over all of these organ systems and how they work together to make your body work. The same information in a different slide right here. A little bit more detail. Kind of like images like this one right here, where you have sort of all 10 body systems and you kind of get a little bit better idea of how they work together and how there's all this interaction. You can see these arrows going back and forth. And other than that, you have also the table view where you have the systems listed, what they include, and a little bit of the function, sort of an overview. Okay, so moving on. The way your book is organized, it typically tries to present the information in at least 2 different formats. So there's very popular in the textbook are these flow charts where you can see one thing affects the next, and so these are diagrams of processes in sequence. And then of course you have the schematic diagrams of structure and function. So there's different ways to present the information, and typically the textbook gives you at least 2 different options, because we all like to learn things a little bit different maybe and have different options of how we like to process new information. So here's an example. It's like a silly example of a flow chart. Doesn't really matter what the content is here. It's just the idea of how these flow charts are organized. So you can take a look at that here. Another flow chart there. This one is more relevant to physiology. So here you can take a look at, here's the issue. Somebody's working out there on a hot day. That sounds familiar these days. And then you're losing a lot of body water, and that leads to body fluids becoming more concentrated. And that leads to receptors that pick up that information and send it to an integrating system. And then all of a sudden you're going to be feeling very thirsty and seeking out water so that you rehydrate your body. So this is sort of a nice flow chart mapping out the sequence of events when you are dehydrated. Let's take a look at function versus process. Function explains the why, and the process describes the how. So we'll talk about various aspects of that as we go through the course. And here's an example. The function of red blood cells is, well, to carry oxygen and deliver that oxygen to the tissues. So the red blood cells, the function of it, they need to have hemoglobin so that they can bind oxygen and then release it to the tissues. Translational research integrates the mechanistic studies and medical treatment. But here, let's take a look at the structure and function theme in physiology. So structure is mostly what you study in anatomy, the parts, and then the function is how they all work together. So there will be molecular interactions within cells, for example, or compartmentation. So you have the intracellular body of the extra and intracellular body fluid compartments, and there is a lot of exchange in how these compartments, there's all this dynamics going on of exchange going back and forth. Again, here there is this very important theme of homeostasis, and that means that's the mechanism that keeps your body functions within a normal range, and we're going to be talking much more about that. So this internal stability. So homeostasis is the regulation of the body's internal environment, and basically you have set point values for everything, whether that is your the concentration of electrolytes that you're supposed to have in your body fluids, or hormone levels, or body temperature, you name it. So everything has a normal range, and homeostasis is that mechanism that keeps you in that normal range. Sometimes the range is very narrow, and it's very carefully monitored. For example, CO2 levels in your body fluids, very carefully monitored. Other ranges are fairly broad, like blood glucose levels. They can go in a fairly broad range, somewhere between 60 to 120 milligrams per deciliter. So you will see as we go through this course that there's homeostasis will follow you, and it's the way of your body to keep you within a normal range. Now, if homeostasis fails, then you're going to end up with disease or some sort of pathophysiological situation. So that would be the study of pathophysiology. Okay. So here is a nice flow chart that kind of illustrates how homeostasis works, and you have this organism in homeostasis up here. Then there's some change, some parameter. Let's say it's really hot outside or really cold outside, and you need to compensate for that loss or the danger of losing homeostasis. And that compensation then is sort of spelled out here. If the compensation fails, then you're going to end up with illness and pathophysiology. If the compensation is successful, then you stay in homeostasis and you feel quite good. Okay, so we're going to take a look at the body's internal environment. You always want to make the internal environment stable. So you want to keep your body in that homeostasis. And we do have interaction between the outside world, and there's exchange also going on within your body, like within the fluid compartments. And we're going to take a closer look at that in another lecture. Here are two different diagrams that try to capture that representation of exchange between the extracellular environment, intracellular environment. So you can see here in this circle diagram or the box diagram over here, you can take a look at these arrows going back and forth denoting the exchange. Now, homeostasis depends on mass balance. Law of mass balance means that the amount remains constant if input is offset by loss. So input has to equal output. And this mass flow, we don't need to pay too much attention to this right now. Take a look at this diagram, and this is kind of better representation of the exchange that's going on. Over here, this is just input equal output, so to keep you in balance or homeostasis. But here, over here, a more specific example of how, let's say with body fluids, you have intake into the body, and then you have output, and overall there needs to be a balance. It needs to be input equal output. We're going to cover this whole topic in much more detail in the gastrointestinal system and also the renal physiology. Anyway, so a little word here about homeostasis versus equilibrium. You might think homeostasis is always equilibrium. That's not true. Homeostasis means that you are within the normal range. Equilibrium means that there is approximately equal amounts of something on both sides of this equilibrium. Let me explain this to you. Here is a very good and very important example of the ion distribution, major ions in your fluid compartment. So ECF stands for extracellular fluid compartment. ICF is the intracellular fluid compartment, and you can tell just looking at it that we have different bars here, and the amounts are not the same. So in other words, let's take a look at sodium first. Sodium ions, there's a much higher concentration of them in the extracellular fluid compartment as compared to the intracellular fluid compartment. That is not an equilibrium, but this is homeostasis. In homeostasis, you must have much more sodium on the outside and a lot less on the inside. Otherwise, your nervous system will not function. Same is true here for potassium, another very important ion. You can tell that the concentration of potassium is much higher inside of the cells as compared to outside of the cells, and that is necessary for action potentials to be able to occur and for your nervous system to function. We'll be talking much more about it, but it's a good idea to know this already, that sodium must be much higher on the outside as compared to the inside, and potassium is exactly opposite, much higher on the inside versus the outside fluids and of the cell, that is. And this would be an example of, if we have this situation here, we're in homeostasis, but we're not in equilibrium. Okay, moving on to control systems and homeostasis. So you can take a look at what's in your book right there. There's not much more I need to say. It kind of works, these control systems, they kind of work like your air conditioning. We're all running air conditioning these days. So if it gets too hot in your house, or in your car, for example, then you want to turn on the air conditioning. So you have some sort of receptor that measures the temperature, and then sends a message to your integrating system, and that integrating system makes a decision. And in the case of the air conditioning, we'll turn on the air conditioner to bring down the temperature in the house back to your set point value, what you wanted it to be. So here are some examples. Here, the input signal, and there's this integration center. That's typically in our central nervous system for the human body, and then you have the output signal response. I'll give you some more specific examples. So here the local control is restricted to a tissue. That's sort of a sidetrack here. But let's take a look at these feedback loops. If you want to be in homeostasis, you need to have what's called a negative feedback loop. It stabilizes you. It keeps you in homeostasis. Positive feedback loops, on the other hand, they will get you outside of homeostasis. And so let's take a look at how this works. Here would be a control system. So you are having a receptor that measures a locally something, and it sends it to the brain that evaluates whatever these receptors measured. And then let's say here in this example, the blood pressure is too high. We're sending a measure. We're sending these measurements up to the integrating center in the brain, and then there will be some compensation to bring the blood pressure back down to what it's supposed to be. Taking a look here at the next slide, you can see that they're using here an example of temperature control. So there's a thermostat that has a set point value, and once the temperature varies from that set point value, a sensor will pick that up and will send information to the integration center, and then we're gonna take the steps in order to bring the temperature back to what it was supposed to be. So you can take a look at these flow charts. In general, the way negative feedback works is you have these set point value. So this is how you stay in homeostasis. You have a normal range, and that is true for all the parameters. So sometimes the range is narrow, sometimes the range is very broad, but it doesn't matter. Your red line, that is the set point value, and then you have the range plus minus from the set point. That's allowable, but you don't want to fall out of homeostasis. That will be falling out of this range right here. So there are normal fluctuations around the set point, but whenever you're approaching the upper or the lower limit, that's when something kicks in and tries to bring you back to the normal range. And then here is another representation of the same thing. Negative feedback is going to be what's driving homeostasis. So you have a stimulus and a response, and you want to stay in that normal range. Now, positive feedback will get you out of normal range. So there are a few examples. Childbirth is the most dramatic one, where getting further away from homeostasis initiates an even larger response, and the whole thing continues in a cycle until that baby is born. And we have here an example of a positive feedback loop that is here shown for childbirth, where more cervical stretch releases more oxytocin, which leads to more uterine contractions, and that leads to more cervical stretch and more oxytocin, and the whole cycle continues and these uterine contractions until the baby is born. There is a BioFlix video that I posted for you that you should take a look at. This one takes a look at the regulation of blood sugar and explains homeostasis, so you should definitely take a look at it. It's pretty cool. It's only a few minutes, so it's not too bad. Okay, a few other things to tie up this whole first lecture. We have something called circadian rhythms, and that is the idea that throughout a 24-hour day, the set point might have fluctuations. In other words, certain parameters, they can have a higher set point in the morning and maybe a lower set point in the evening. And so that means even the set point values, the normal ranges, can have variations. And so here is diagram that shows you I like the plasma cortisol concentration here. So this here is your stress hormone. If you have cortisol in your blood, you have a little extra sugar, you get going, you're active. So not surprisingly, the cortisol levels are higher in the morning when you're active, when you just got up, and then when it's dark in the evening, they should be lower. You might have noticed if you're really stressed out and you have very high levels of cortisol in circulation, it's going to make it very, very hard to sleep. So if the cortisol levels are too high, it's going to be hard to fall asleep. Cortisol is supposed to be high during the day when you're active, and it's supposed to be low to help you fall asleep in the middle of the night right there. Okay, now moving on to experimental design and scientific inquiry. This is mostly stuff that we also cover in the lab, so I'm going to keep this very short. We will be talking about the scientific method in the lab. So be sure that you're going to take a look at those exercises that you're supposed to be doing. graphing, that is something that's very important in science in general, and in physiology in particular, we deal a lot of times with bar graphs. The reason for that is that we have groups, and in physiology, most of the time, we're going to take a look at one variable, and in order to figure out whether there was an effect or not an effect, we need to study groups, because in physiology, we're dealing with living systems, and not everybody is the same. And so we need to have groups of people. Think of the following situation. You have a new medication, you want to test out if it's effective. Or what they're doing right now was testing out the COVID-19, the vaccine. So you have a control group and you have an experimental group. You want to vaccinate your experimental group and not vaccinate the control group, and then see if the vaccine was actually able to reduce the number of infections compared to the control group. So now then you collect your data from the groups, and then each group is represented as a bar in a bar graph, and then you do statistical analysis on comparing the data and see if there's a significant difference, whether or not your vaccine worked, in other words, or not, or your new medication, it's effective or not. Scattergrams like this, we're not going to be dealing too much with that, but if you have data, single data points, you can see at least trends with a scattergram like this, but bar graphs are actually better for physiology. So here will be a good example of a bar graph. Here in this case, you have 3 groups, and you want to compare the effect of some diet on canaries, I guess, here. But the point is, each bar represents a group, and it's the average of the group. So the height of the bar is the average within that group, and then you're comparing the groups to one another. Now, if you have continuous data that change over time, usually, then you can make a line graph. So you take your samples, your measurements, and each one will be a dot on a line graph, and then you can connect the dots because the in-betweens, they all exist. And so you're allowed to make the connections because you have a continuous change. So line graphs are great for continuous change. Now here's an example where you have individual data, and you can see there's all sorts of variation, and in order to find sort of a trend line, that is something that can be done with statistics, where you can just do a regression analysis and see if there is a sort of you will find a good correlation between your data points, as here in this case, time spent studying, does it have an effect on your exam score? And so you can have your data points there, and then you find a trend line, and looks like actually studying works. So nice hint right here. And our final slide for this lecture, the interpretation of human experiments. Sometimes they can be difficult to interpret, and we will talk about this more in the lab as well. Whenever you're dealing with humans, with individuals, there's different responses, and so you can even look at COVID-19 right now. People respond differently. Some get very sick, some die, and others they hardly have a sniffle, and people react differently to medications, to pathogens, to whatnot. So sometimes this variation can make it sometimes difficult to interpret human experiments or human data. So there is a lot of genetic variability, environmental variability, and that's why people also react different to different medications and different pathogens. When you're doing a study, you definitely want to have always a control group and an experimental group. And let's say you are testing a new medication. If you tell people that are in the control group that they're not getting anything, then they won't report anything because they know there wasn't supposed to be an effect. So it's more effective, and you get better data if you're using a placebo study, because when somebody takes something, there's this feeling, yeah, this should work, or this should do something. So if you're giving people that are in the control group, you're giving them a placebo, it's like a fake, it's a look-alike, but they still take something, and so they might report some side effects or something, and then you can compare that. You get better data for comparison with your experimental group. And something that's actually been in the news lately, the ethics of humans as test subjects. They are running a study right now where they are testing a new vaccine, a potential candidate for vaccine for COVID-19. And they are using that vaccine and then on purpose infecting these people after, I think, 2 weeks or so after the vaccine should be effective. Then they infect them on purpose with the virus to see if the vaccine is effective. Now, that's kind of a shortcut to the normal protocol, the normal procedure. You're not supposed to do that. And some people have raised some ethics concerns. But on the other hand, we are in such desperate need to have a vaccine that they're kind of doing some shortcuts. And of course, these volunteers, they were informed that they will be infected with COVID-19 and hopefully they'll be doing okay. But this could also easily be not so okay. Anyway, that's it for this introductory lecture. And please take a look at the slides, maybe the textbook if you want. And also definitely now you need to go take a look at the lab.